Hello friends, we are discussing about MRI brain, its principle and differences between T1, T2 and FLAIR images. The principle is to say simply give energy to, because the protons will be arranged randomly in the normal cells. So if you give energy, it excites the protons, then suddenly turn off the energy. Third, if you give energy, the proton will be excited. So if you turn off the energy, the excited proton will come to again back to the previous before energy giving state. So the energy will be, we get back that energy, that is called rate of relaxation. So simply say give energy, off the energy, then observe the energy which is gotten back after the protons are released. Then main difference between T1 and T2 images. In T1 fat is bright and what in T2 is dark. Water is dark in T1, white in T2. Then as fat is bright, we can see fat containing structures more clearly in T1. Then coming to flare. Flare simply T2 plus CS of dark, because by making CS of dark we can see any edema or structures close to ventricle, because CS of white and structure the edema structure close to is white, we can't differentiate. So to make this, we made the other sequence called flare. Now see the difference between T1, T2 and flare. In T1, CS of dark, CS of dark, bright, dark. Okay. Then coming to white matter. White matter is which is in the center. Cortex is cortex. So in T1, you can see in T1 weighted images, you can see I am going to mark now. The cortex is gray. The cortex is gray and the central part white. Light is white matter. And T2 light gray and dark gray. Then coming to diffusion weighted image. In diffusion weighted image, because of diffusion restriction, this is ADC. Now again coming back to what is diffusion weighted image. In diffusion weighted image, usefulness is it can detect early ischemia in the cells. Then what is diffusion restriction? So in this the principle is it recognizes the fluid restriction as bright. How the fluid restriction occurs? In early ischemia, because of energy failure, in the cell wall there is sodium potassium ATPase pump. I write sodium ATPase pump, but there is sodium potassium ATPase pump. So because of energy failure, sodium potassium ATPase pump will not work. So sodium will be remain in the cell. So sodium takes the water. So it causes cytotoxic edema. So fluid will be remain in the cell only. So it causes fluid restriction in the cell. So it is, so we can see this as bright fluid restriction. So the image we see that white color is in DWA is ischemia. Then we have come to ADC. This is dark. So white and dark in ADC.